Hi, my name is Mallika, and for our DMD project, my teammate Freddie and I decided to interview a researcher to understand a different side of DMD. But before we get into the interview, let's go ahead and meet both of us, and then we'll get into the good stuff.
Hi, my name is Mallika. I'm a fourth year physiological sciences student and I'm applying to medical school this May. And so what was really interesting for me is I really like seeing how different organ systems work together. And so I read a paper by Dr. Rinaldi that talked about how the suprachiasmatic nucleus actually combines with something like DMD. And so what I was really interested in was asking him more questions about how two seemingly very different topics or parts of our body actually come together. I was really interested in learning more about his creative tools in his research. So yeah.
Hi, I'm Freddie. I'm a senior majoring in MCDB. I'm actually an incoming PhD student, so I'm going to be starting this coming fall. And I have a real strong interest in cancer cell biology as well as pharmacology. Dr. Rinaldi was actually a perfect fit for us as we were really keen to better understand the experience of a researcher in tackling muscular dystrophies such as DMD. My interest specifically in pharmacology led me to his paper Antisense Oligonucleotides, the next frontier for treatment of neurological disorders. And in this paper specifically he talks about various treatments such as ASOs as ways of perhaps treating DMD. A current theme in cancer is combinatorial medicine and how powerful it can be. And his paper specifically explores different ASOs. Our kind of story that we were interested in specifically was the potential of combinatorial medicine for muscular dystrophy, perhaps a combination of different ASOs or an ASO with a different treatment. We therefore interviewed Dr. Rinaldi to kind of elucidate this process and this question. I'm delighted to introduce Carlo Rinaldi, who is Professor of Molecular and Translational Neuroscience at the University of Oxford. Dr. Rinaldi completed his medical training at the University of Federico II in Naples, Italy. He then moved to the NIH for his PhD on pathogenic mechanisms of spinal and bulbar atrophy. And in 2015, he translocated to Oxford as a clinical research fellow. He now holds a prestigious UKRI MRC Senior Clinical Fellowship, and his research focuses on the identification of genes associated with neurological diseases to increase understanding of the molecular basis of these diseases and the development of new treatments for hereditary neurological diseases. We are very grateful for you, Dr. Rinaldi, for sparing the time for this discussion today.
Very pleased to be here, Freddie.
Okay, so I'm probably going to start us off with just some introductory questions, get to know you and your research at a general level. So if you could start off just by telling us more about why you chose to go into research. And I know you mentioned you're not a directly focusing on DMD, but why is that part of your research and what's interesting to you about muscular dystrophy?
Well, I mean, first of all, I should say that I'm an adult neurologist, and this is how initially I started my journey in medicine. So as a 13, 14-year-old, I thought that one of the coolest things that I could possibly do was to study the brain. And I had very clear in my mind that I wanted to become a neurologist. And this was so clear to me that actually very early on in my studies, I knew that that's the only thing I wanted to become, a neurologist. I wasn't interested in anything else, effectively. My drive, in a way, into research was kind of like comes from the fact that in neurology, you can, particularly in neurogenetics, which is the field that I was working on initially, you do a lot of you give a lot of very clever diagnoses to patients, which are deadly diagnoses most of the time, and there's nothing else that you can do. So you follow the patient, every 6 months or every year, you see a slow or fast deterioration, and there is effectively very few treatments for these patients. So my initial motivation, which remains the motivation to this day, and the reason why I wake up in the morning and I stay up at night, is that I want to develop treatments for patients. And I want to develop treatments particularly for rare disease patients. So patients that have rare diseases that very few people know about, and think that there is no hope for them. This is what we're trying to do in the lab. So I built a team now of researchers working on rare disease, understanding mechanisms of diseases, and how we can deploy and use this information to effectively develop gene therapy treatments and clever treatments for these conditions. I spend my time between the clinic and the lab. I'm in the clinic once a week, and the rest of the time I'm in the lab. And I'm very proud to say that I'm surrounded by a very passionate group of people. We share this goal and this vision of treating patients.
Yeah, thank you so much for sharing. I love that. I'm curious, you were 13, 14. What did anything spark that, or did you read something? Like, how did you come across neurology?
I mean, I thought I was very fascinated always by things that are unknown or mysterious, and of all the things, I've always thought that the brain was the most interesting of all the things. So it's I don't know. I just felt like it was my call. It's just my thing. I want to understand how our brain work, and also I would say I'm a very I want to say humane person. I mean, I like humans in general, so I want to understand how they work, and probably that's my primary motivation.
Yeah, I hear that. Well, thank you for sharing. Okay, so in terms of, what ongoing research is happening right now, apart from what you're doing, are there anything that's kind of out on the horizon that's looking particularly interesting? Are there any new breakthroughs that you think that we should keep our eyes on about related to muscular dystrophy or just, neurology in your field specifically?
Well, I like very much the idea of using gene or genetic regulatory information to understand how a gene is switched on and switched off. And I think this is kind of like surfacing as a very rich area that you can then tap on to develop treatments effectively. So if you learn and if you understand how a gene is switched on and switched off in different cells, in different tissues, in different conditions, then you can develop a treatments to force the system in one direction or another, depending on your need. And probably at the moment, I would say gene therapy of all the tools around gene editing are probably the most exciting thing, and how we can push them into the clinic. This is going to be the challenge probably of our generation. So currently in the lab, we're using a lot of gene editing tools to screen for regions of the genome or of a specific gene that we want to downregulate or upregulate or splice in, splice out. And yeah, that's probably the thing that is, I would say, is the most exciting at the moment.
Got it. And what are you working on right now to upregulate or downregulate?
So we're working on two main themes. So the first is to look at enhancers. So enhancers are region of the DNA that loop onto generally promoters of gene and enhance, as the word says, the expression of a specific gene. And this region can be very far away from the promoter of a specific gene. So we want to map those regions, understand where those regions are. We're doing this in the context of skeletal muscle and motor neurons initially. And the idea will be to, again, trying to force this system or disrupt this system in case a disease is linked to reduced or increased expression of a specific gene. And the other theme
Got
in
it.
the lab is to work on RNA editing, which is a modification of RNA that occurs naturally. And we want to understand what are the rules of RNA editing, because again, by doing that, we might want to exploit this information to again develop a system to use an endogenous way, let's say, to edit the transcriptome to force a mutation or correct a mutation that will rescue a disease effectively.
Yeah, very interesting. Okay, got it. Yeah, thank you so much for expanding. And then lastly, just are there any aspects of DMD or muscular dystrophy, for example, nutrition or sleep? I know I read a brief paper about that you wrote where you kind of dove into sleep and the clock genes in our SCN and how that relates to muscular dystrophy. So maybe are there any aspects that you think are overlooked currently in muscular dystrophy research that should not be, that you think need to have further studies?
Well, one of the very apparent areas that are becoming interesting and more and more studied, let's say, in the context of Duchenne muscular dystrophy, is the fact that this is a very much of a multisystemic disease. I mean, it's certainly primarily affecting muscle. I mean, as the word says, muscular dystrophy. But in reality, patients often have other, symptoms of other tissues involvement, heart typically, but also the brain, and restoring Duchenne or dystrophin expression in those tissues is going to be critical. So one interesting area of development is the fact that you don't want to restore dystrophin exclusively in muscle, but you might want to restore dystrophin also in brain. For example, many of these kids will have also a phenotype related to the lack of dystrophin with reduced cognition and even autism spectrum disorder. So restoring those isoform might be essential if we want to really cure this disease. And this is becoming an interesting area of investigation, I think.
Yeah, very interesting, and I'm sure very relevant to what you're interested in, well, in terms of neurology in our brain. So, okay, well, thank you so much for answering some of those questions. I'm going to turn it over to Freddie, who will ask into some deeper paper-specific questions.
Yeah,
Thank you.
so some of the kind of current mechanisms that you're talking about for potentially treating various muscular dystrophies or neurological disorders sounded really interesting, especially kind of RNA editing via some sort of endogenous molecule. That sounded really cool. Um, in class, we've been talking quite a bit about exon skipping specifically and its potential role as a therapy. And I read one of your papers, Antisense Oligonucleotides, the next frontier for treatment of neurological disorders. And it was very relevant to kind of what we were covering in class. A really interesting read. And I just have a couple questions that I kind of conjured up, which I was just wanted to ask you about, just so we could get a
Go for
broader
it.
understanding of various kind of specific aspects of it. So,
Yeah.
of course, exon skipping produces a truncated dystrophin protein. And although the N and C terminal domains are still present, there are always going to be some domains, potentially important ones that are missing. How stable are these truncated proteins in the cytoplasm? And would patients need this sort of antisense oligonucleotide treatment every few days, every week, kind of what would be the timescale for it?
Yeah, it's a good point. I mean, actually, for many of the questions you just asked, we don't have an answer. So we don't know exactly
Hmm.
what is the dynamics of this truncated form. I mean, most likely this truncated form or this mutant, let's say, dystrophin transcript, they will have a limited half-life in the cell. So they will undergo nonsense mediated decay. So the point of exon skipping is to restore the reading frame in such a way that there is a complete or finished product that is produced and can be translated into a protein that will then eventually stick around and potentially substitute, let's say, or replace the dystrophin that is lacking. Typically, patients don't need daily injections. I mean, the good thing about antisense oligonucleotides is that they are quite stable. They are quite stable in our body. They can get attacked by nucleases in our system, but once they penetrate the tissue, they tend to actually stick around. And the other component to that is that dystrophin itself, as a transcript and then as a protein, has quite a long half-life. So once dystrophin is produced as a protein, it has a very structural role, and then it takes days before there is a full turnover of that specific protein. So usually in the ASO field, patients would require treatments every few months rather than weeks. It is a repetitive injection, and of course, delivery is a main issue of antisense oligonucleotides. You want to make sure that the tissue that you want to target is, in fact, reached properly. And the issue that you might understand of skeletal muscle is that it's extremely abundant.
Yes.
Skeletal muscle is about 40 to 50% of our body mass. So you need really a lot of antisense oligonucleotides because technically in DMD, patients lack dystrophin in all tissues effectively. So this is one of the issues that the DMD field is indeed facing. So that you need a lot of ASO and ASO that is able to penetrate skeletal muscle.
Yeah, no, that was definitely something that's also been highlighted in lecture as an issue. Thank you.
Yeah.
Um, Another thing that been covering in class that you also mentioned in your paper was the drug etiplacin. I probably said that very incorrectly, but the data showed that it didn't seem to have too much benefit to dystrophin levels, which increased by around 1%.
Yeah.
And I also saw that there were various other drugs that have also been used in clinical trials,
Yeah.
um, such as Pro 045 or SRO 4053. Has anyone ever tested whether a combination of two or three of these drugs would be more effective than one alone?
It is a good point, and I will probably and you're right in saying that most of the clinical trial data that are available so far in the context of DMD, they all rely on very little improvements, let's say, very little augmentation of the dystrophin protein. I mean, I have to disclose also that as a community, we have also we really don't know how to measure dystrophin effectively in muscle. Muscle biopsy is often required, and taking a muscle biopsy from a tibialis anterior, for example, of an individual as opposed to a biceps. You have to assume that that's a proxy of what happens in all the rest of the muscle, but in reality, that might not be
Okay.
the case.
Yeah.
And in many clinical trials, what you do is that you take a muscle biopsy before the treatment and after the treatment, and you try to quantify based on immunofluorescence or Western blot, try to quantify the amount of protein that you see under the microscope, and making an assumption, inferring that levels is what happens in the rest of the body. But in reality, we really don't know if that's the case. Your question about the combination of combinatorial medicine is certainly a very, very important one. And I would say that in the context of neurological, neurodegenerative diseases, combinatorial medicine is probably the way to go. And I feel like that
Mhm.
in the cancer field, probably our colleagues got that right much earlier. I mean, there's usually never a single drug or a single molecule or a single treatment that treat cancer patients. While we in the neuroscience and the neurology field, we're still stuck with this idea, that there's a single molecule will treat such a complex disease, and such as Duchenne muscular dystrophy. But most likely, the reality of things would be that we will need more than one molecule. And I'll give you probably a better example, which is not Duchenne muscular dystrophy, but spinal muscular atrophy or SMA, which is in many ways a much more successful story when it comes to antisense oligonucleotides or therapy development in the last, let's say, 5 to 10 years. So we have oligos or ASO or gene therapy that works very, very well. So they effectively save the life of these kids. But more and more, we're learning that combinatorial treatments can even can push this ameliorations even further. We just had a paper out about last year where we used a combination of an antisense oligonucleotides such as nusinersen in a mouse model of the disease combined with a small molecule, and we demonstrated that by using the combinatorial medicine approach, we were able to effectively have a better outcome, reduce the dose of the ASO that we had to use, and reduce also the intervals of the administrations. And this has important implications because ASO treatments can also be very expensive. And
Yeah.
of course, reducing the dose of an ASO and the administration can have serious clinical impact.
Wow. Okay. Very interesting. And that kind of answered my final question that I was going to ask about whether this sort of combinatorial medicinal approach holds a lot of potential or therapeutic benefit in the future. And that's really cool. And that's a very interesting paper. I think I'll probably be in touch with you after this if you could send it to me. That would be a really cool
Sure,
read.
sure,
That
absolutely.
that's all of my questions as well. Thank you so much.
No problem.
After meeting with Dr. Rinaldi, we did some of our own digging online to see what combinatorial therapeutics could look like. And a few papers interestingly looked at both utrophin upregulation and dystrophin expression in conjunction. An interesting potential concern was that since both dystrophin and utrophin anchor to a bunch of complex proteins in the sarcolemma, like the DGC, that there would be a finite number of slots, as in there would be competition to bind. However, the research has actually proved that both dystrophin and utrophin can co-localize, meaning that they can occupy the sarcolemma at the same time. And so that even with this with these limited binding sites, co-expression does not interfere with membrane localization. And so, very importantly, the combinatorial therapeutics improved muscle function better than either therapy alone. And so this suggests that actually the number of binding sites is not actually so limiting to prevent combinatorial therapeutics from functioning. And it's also possible that in the wild type model, there may be unused spots. So then now in this case, we're actually able to maximize the number of spots for binding for both dystrophin and utrophin. And so it's really interesting, as Dr. Bernaldi says, it really points to the future as possible therapeutic aims for DMD research. We had the privilege of interviewing Dr. Bernaldi, a physician scientist at Oxford University Medical School, specializing in neurological disease. His passion for neurology began in his teenage years, sparked by a fascination with the complexity of the human brain. Motivated by a desire to help others, Dr. Bernaldi pursued a career in medicine, seamlessly blending a love for his research with a strong commitment to patient care. We asked Dr. Bernaldi about recent advances in the treatment of Duchenne muscular dystrophy, or DMD, and other neurological disorders. He highlighted gene regulation, specifically the ability to switch genes on and off by targeting gene enhancers, as one of the most promising areas of research. Another innovative approach being explored in his laboratory involves the control of RNA editing. Both strategies aim to develop small molecules capable of selectively activating or silencing gene in specific tissues. While the foundational science is advancing rapidly, Dr. Bernaldi noted that the translating gene editing into a viable human therapy remains one of the defining challenges of this generation. Discussing DMD specifically, Dr. Bernaldi reflected on exon skipping, which is a technique that's shown promise in theory, but has delivered underwhelming results. He suggested that a combination therapy involving 2 or more drugs might yield better outcomes, much like the transformative effect it's had in cancer research. However, he observed that neurologists have been slower to adopt such combinatorial approaches, remaining more focused on single drug therapies.
Dr. Rinaldi also pioneered the use of stable ASOs for the treatment of neurological disorders. In DMD, the most common mutations disrupt the open reading frame of the dystrophin gene, with large deletions accounting for nearly 70% of all identified mutations. By modulating the pre-mRNA splicing of dystrophin, ASOs can restore the open reading frame, offering a potential therapeutic pathway. Despite these advances, significant hurdles remain. Around 40% of the human mass is composed of skeletal muscle, presenting a major challenge in delivering sufficient quantities of ASOs to affected tissues. Moreover, DMD impacts not only skeletal muscle, but also the heart and the brain. A key goal for future therapies will be achieving targeted delivery to specific tissues at levels sufficient to restore functional dystrophin expression. Although great strides have been made in understanding the genetic basis of DMD, further work is essential to develop effective tissue-specific treatment for use in patients.